Right, good afternoon, everybody. Thank you for joining us for the fourth in our series of ESHG webinars. I'm Bill Newman. I'm the immediate past president of the ESHG, and we established this webinar series early this year to showcase some of the wonderful genetic research that's going on across the world, both in Europe and further afield. And today, that's a perfect example in place related to that. So it's my absolute delight and pleasure to introduce as our speaker today, Kaitlin Samocha. Kaitlin is an honorary European, as she spent plenty of time in the UK as part of her illustrious career so far. So she did her undergraduate studies in Chicago before doing her PhD at Harvard, and then moved to the Sanger Institute in Cambridge, and has been part of the ESHG Scientific Program Committee, but more recently has set up her own group at the Broad Institute and has been part of the team there that has really advanced our understanding of variant classification and the value of population scale data through her work on the Nomad database with a number of her colleagues. And so Kaitlin's used that information to understand the pathogenic nature of variants related to a whole range of different conditions, and how we can use that data to understand which variants might be benign and which ones might be pathogenic. So it's absolute delight to have Kaitlin join us and share her expertise this afternoon. So thank you, all yours, Kaitlin.
Thank you so much for the intro. It's always an honor to present, and I appreciate the opportunity to be able to show off some of the work that, as you mentioned, a large team has been contributing to. So one of the primary goals of medical genetics is to be able to identify genetic variation that influences a given trait or disease. And disease is a lot of the focus. But one of the primary challenges that we're running into is not necessarily the identification of the variants, especially for small variants. We're quite good at finding them now. The issue has become the fact that we have millions of genetic variants in any individual genome, the vast majority of which are neutral. So this is quite an old figure from the 1,000 Genomes Project, but you can see 4 to 5 million genetic variants for populations around the world, with European populations colored in blue over to the left, and African populations colored more in the orange and yellow off to the right. And I will argue, like many have argued before, that to understand the variation that we see in an individual person, let's say a patient, we really need to study the variation found in the population. So we want to study these patterns across all of these individuals from around the world. And the way that these can help are in multiple ways, but in two primary ways that we'll discuss today. So we want to use this population data to help us come up with ways to prioritize potentially damaging variants in a patient. So if you're in a clinical setting, obviously phenotype is quite important, but we're going to be studying the genetics today. So off to the right, we have variant level evidence. For example, focusing on genetic variants that are predicted to be deleterious based on a functional consequence, potentially some in silico score or a variant effect map that has been made by functional analyses in a dish. Of course, the presence or frequency of that variant in the general population or a reference database is extremely powerful information. On the gene level or region level, you can also start to prioritize based on whether the variation in that gene is already associated to disease, potentially expression in relevant tissues, and of course, whether that gene seems to be tolerant or intolerant of mutations, again, in this general population or reference population. So if it's not tolerant in the general population, it might be more likely to lead to severe disease when it's disrupted. So just so we're all on the same page about nomenclature, we have some idea, at least in the coding region, what might be more deleterious. So if, for example, an AAA codon makes lysine, there are a few mutations that could lead to synonymous, so the protein's unchanged. Missense mutations are, for example, AAA becoming ACA, and now it's a different amino acid. However, it's this class of mutations off to the right that are typically considered to be the most deleterious. So stop codon, for example, a premature termination, a frame shift that changes the frame of the rest of the protein and often leads to early termination, or canonical splice site disrupting mutations that impact the ability to splice together different exons in the gene. So throughout this presentation, I'll try to be good about referring to these as putative loss of function or P-loss variants, but sometimes they're referred to as protein truncating variants since they often truncate the protein prematurely. So given some of this knowledge, we're still talking about quite a lot of variation in an average genome. So there's that 4 to 5 million variants I mentioned off to the side, the vast majority of which are single base changes or short insertions and deletions. I should note larger structural variants impact more of the bases individually since they're so large, but they are not that many. We do see about 70 newly arising de novo variants per person. As we start to focus on these functional classes, we're seeing 10 to 12,000 synonymous variants, 10,000 missense variants, 150 of those PLOF variants, and even up to 50 reported disease-associated mutations from databases like ClinVar. We can further filter these, for example, by using a reference population. And what I'm showing on the bottom is the rare variance across populations, and this is an allele frequency cutoff of 0.1%. And we can see rare PLOF variants, we're still talking about something like 10 per person, or 150 rare missense or indels per person. So even with these filters, we're talking about a lot of variance. So we've taken it down from 4 to 5 million to 10 to 100, but that still can be quite a lot to sift through if you're trying to find the single variant or combination of variants that are contributing to a disease. However, this is an excellent time to be doing genetics because our databases are getting even better. And I hope I'll convince you throughout this talk that the larger and more diverse the database is, the more useful it is in these types of analyses. So we have seen increasingly large sequencing datasets of reference populations over the past decade or so. I used to show this plot with many more datasets, but here I'm only focusing on those that are over 100,000 individuals, and these are, again, publicly accessible. So we have the TopMed data, about 132,000, the Genome Aggregation Database, or Nomad, which is what I'll be focusing on. So we had a V2 release a couple of years ago of 141,000. The All of Us database is currently at 245,000. UK Biobank, of course, around 500. And then Nomad V4.1, which we'll discuss, around 800,000, and their Genome Genetics Center million exome dataset, a little over 800,000 as well. You'll see across all of these bars that they are colored, and the coloring here is by genetic ancestry group. So this is really based on genetic similarity to individuals that have been pre-labeled. You'll see there's quite a lot of this lightest blue. So that indicates individuals of a broadly defined European genetic ancestry. And I really just want to emphasize that none of these databases are globally representative. There are far too many individuals coming from this European genetic ancestry background. And as you can see, for example, not that many individuals from a Middle Eastern genetic ancestry backgrounds. There are a few individuals both in Nomad and the Regeneron dataset, but it's only a couple of pixels down here. So clearly these databases have some work to do. We're aware on the Nomad side of getting better representation around the world. And this is just a bit of a warning. So if you're coming to these databases with a patient who's not well represented, there are certain caveats you might want to keep in mind when you're looking at this, just that your individual might not be well represented in these databases. So we're going to focus here on the Genome Aggregation Database, or Nomad V4, specifically the 4.1 release. This is 731,000 exomes and about 76,000 genomes that were all natively aligned to GRCH38. The vast majority of these samples are coming from case control studies of complex adult onset diseases, as well as from biobanks. So we have over 100 studies around the world, over 300 data contributors. We do remove related individuals, and we try to remove projects where the child, for example, was selected for a severe pediatric disease, but these are not perfectly healthy individuals. And this is why we call them reference populations. No one is perfectly healthy, just to be honest. However, this is supposed to reflect the general population. So we do have individuals in this database, for example, that have certain cardiovascular traits. And we don't actually see for a particular enrichment of any given indication or disease type, but we have some information on our stats page about that. But we just really want to emphasize that these are reference individuals. They're not perfectly healthy. And we do see about a 50-50 split between XY and XX individuals. So what can we actually do with 800,000 individuals, and how can that help us with variant interpretation? So obviously filtering variance is the most important and powerful piece, not just by allele frequency. We'll also talk a little bit about curation. So clearly one of the most important uses of these reference databases is to be able to apply BA1, BS1, or BS2 for those that know the ACMG code. So this is removing variants that are seen too commonly to be causing disease. So for example, when we took individuals of a variety of genetic ancestry backgrounds and filtered them to 0.1% based on a 6,000 individual cohort, this is actually the NHLBI's exome sequencing project cohort, we found is that there were 600 to 1,000 remaining variants to filter through. And of course, as you jump up the sample size, this is using the 60,000 individuals in the Exome Aggregation Consortium project, we saw a 5-fold reduction. So that's a 10x jump for a 5-fold reduction. We also should note here that this larger data set was more ancestrally diverse in terms of genetic ancestry groups. So that's partially why you see more of a clustering, less of a spread of individuals here. But this is massive. This is much more helpful when we're doing one of these arbitrary filters here. So given that this is important, we wanted to kind of visualize what we see as we move from the V2 dataset to V4. So I'm showing you the number of individuals in Nomad V2 on the left and the number of variants with this 0.1% allele frequency on the right. So these are the ones that you'd filter out. So Nomad V2, you could filter out 500,000 variants just based on that data set alone. And as we move to V4, you can see a massive jump in the sample size. I'll just end here. But the number of variants with an allele frequency greater than 0.1 doesn't actually change that much, specifically for the European genetic ancestry group. So just to see that again, as this jumps up 5, 6, 7 times, actually we're seeing not that many more common variants coming specifically from the European genetic ancestry group. So just to reinforce here that, for example, having individuals of different genetic ancestry groups allows us to find variants that are common in those specific populations, and those can now be removed as candidates for review. But how common is too common for a variant to be pathogenic? Obviously, as you move down the allele frequency spectrum, and this is a figure from a 2017 paper, you're seeing fewer protein altering or so peel-off or missense variants per person that you need to evaluate. And I've thrown out some arbitrary numbers, 1%, 0.1% minor allele frequencies, but these are all arbitrary. So work from this paper and other population genetics suggests that causal variants for severe diseases should be much, much, much rarer. So you can just use allele frequency cutoffs in Nomad of 0.1%. You could use presence absence, though we don't necessarily suggest that, given that sometimes there are errors in the database. But you could also, for example, use a more complicated filtering approach. So one thing that we provide on the website is a filtering allele frequency. So this is a statistical adjustment around the highest genetic ancestry group frequency. It's mostly just to account for the fact that we're sampling from populations around the world. So we take the highest group specific Nomad count and we pre-compute this filtering allele frequency. It's just sort of a bound around that. And then if you have information about the genetic architecture of the disease you're studying, you can come up with a disease-specific maximum allele frequency you might expect to see. So for example, you need some information about disease prevalence, maximum genetic contribution, penetrance. Of course, that's always quite hard, but you can use that information together to come up with this population frequency. And I should say for things like hypertrophic cardiomyopathy, it's actually much, much rarer than 0.1%. It's something like 10 to the minus 5. So with these two pieces of information, so the genetic architecture informed population allele frequency and the filtering allele frequency, this is a more disease-specific way of filtering. However, I do want to really emphasize that when you're looking at databases such as Nomad, but this includes UK Biobank and all of us, the more interesting something looks, the more likely it is to be false. So for example, if you see a variant in Nomad that you don't think should be there, we suggest that you look into the data. So on Nomad, we provide quality metrics and genotype metrics so that you can decide whether this looks like a real variant. For example, the depth, the allele balance of heterozygotes, site quality metrics. We also provide IGV screenshots. So here, this is actually the Delta F508 deletion, infirm deletion from CFTR that I'm showing you. And you can see this 3 base pair deletion nicely in the reads, and you could scroll down and see others. The reason we provide some of these raw reads is that sometimes you can find homopolymer runs or complex indels, poorly aligned regions, just something that makes you suspicious that this is a real call. And this is part of the reason that we say seeing a singleton in Nomad doesn't necessarily mean that you should use that as evidence for ruling something out in a patient, because there are errors sometimes in the database, particularly in these kind of complex regions, these homopolymer runs or regions with segmental duplications. We can start to see a lot more variation there. It's both enriched for real variation as well as enriched for artifacts. So this is one thing to keep in mind, that some of the data in the data set could be an error, though we try very hard to clean it up. Another aspect is that not all variants do what they say on the tin. So for example, loss of function variants, in theory, should all lead to the loss of the function of that protein, but we have done some manual curation, I should say Anna Donaluria's team's been leading this, and can establish that not all of them seem to be acting this way. So the article on the bottom is a framework that they developed to manually curate PLOF variants. And we see when we manually curate homozygous loss of function variants in Nomad V2, 28% of them don't look like they're loss of functions. And when we look at heterozygous loss of function variants in 22 recessive genes, 27%, so a very similar percent, look like they're not loss of function. If you look on, for example, V2, we do actually have a loss of function curation column there that gives information about why it was categorized as not likely loss of function. So for example, in GBA, this variant I'm hovering over is in a homopolymer run. There's some mapping issues. And so this is a variant that is considered to be a pathogenic variant in ClinVar, but we have some suspicions about it from this curation framework. So this is work being ongoing. We see about, again, this 30% or so that are not loss of function. But we wanted to start to update this in V4. So if we look at Nomad V2 on the left and Nomad V4 on the right, we are seeing that there is an increased representation of ClinVar variants in the V4 database. So in particular, maybe it's not surprising that the benign, likely benign column here is moving from 70% to 83%. We did massively increase the sample size between V2 and V4. But we're also seeing that pathogenic, likely pathogenic, we saw 16.3% of them in Nomad V2, and now we're seeing 30%. So a nice thing about this, maybe it's a little concerning to see all these ClinVar pathogenic variants in Nomad, but most of those pathogenic variants have lower allele frequencies in V4 compared to V2. Again, sometimes people make it into these databases or make it into biobanks and therefore are represented in this data set. Sana, who led this work, actually wanted to dig into this a little bit more, and unsurprisingly, most of those pathogenic variants from ClinVar that we're seeing in Nomad V4 are associated with autosomal recessive inheritance. So this breakdown on the left here shows you the pathogenic and likely pathogenic variants in ClinVar. Yellow is recessive, orange is dominant or recessive, red is dominant, and black is X-linked. So the proportions totally in ClinVar, we see that compared to the pathogenic, likely pathogenic breakdown in Nomad, we see an enrichment specifically of these recessive associated, this yellow bar here. However, we do see some pathogenic variants associated with autosomal dominant genes and particularly severe conditions. For example, there are 13 variants in KMT2D that are pathogenic or likely pathogenic in ClinVar that we see in our database. And we have been digging into that a little bit, and it looks like the vast majority of these are coming in from biobanks. So a big contributor, for example, is UK Biobank, which makes up approximately half of the V4 release itself. So if someone was healthy enough to get into UK Biobank, they're now being represented in our data set. And this is something, again, to keep in mind. No one is perfectly healthy, but we do see a handful of people in our database now with what we would consider pretty deleterious variants for severe pediatric disorders. Of course, penetrance is always something to keep in mind. That is a different bugbear that we won't go too much into in this particular talk. So digging further into this, Sana wanted to look at for example, some of those variants that you might not trust in this data set. So they did a deep dive into 734 loss of function variants in 77 haploinsufficient genes. So these are just from the genome sample. These are genes that you would not expect to see loss of function variants in this data set. So we applied that loss of function curation framework, and to shorten the story, 70% of those seem like they do not actually result in a true loss of function effect. So on the right here, that 27, 29%, those are the numbers I was showing on the last slide, though I should little differences. So again, recessives about 27% seem like they're not necessarily causing loss of function, but for these loss of function variants in haploinsufficient genes, the majority of them seem like they're not actually real loss of functions. And the reasons for that are on the left. So some of them are in the last exon, some of them have a frame restoring indel, but a lot of them are a combination of different variants. So again, if you see something in the data set that seems suspicious, it might not be real. But this is one powerful aspect. So you can filter your variants based on allele frequency, but you can also keep in mind some of these curation efforts. So beyond single variant filtering, we can also look at the patterns of these mutations to identify genes and regions that seem to be intolerant of those mutations. And this is called selective constraint. So using the patterns, we can define genes that are intolerant. There's two primary ways of going about this. So the first is a shift in the allele frequency spectrum. So let's see that we see more rare variants than common variants in a gene. So we just don't see common deleterious variants. And the second is depletion of rare variants compared to an expectation, potentially coming from a mutational model. So the first of these, the shift in the allele frequency spectrum, is best characterized by RVIS. So this was led by Slavoj Petrovsky and David Goldstein. It's a way to identify genes with less common functional variation than anticipated. So what I'm showing you on this plot here is from their 2013 paper. And on the X, you see the sum of all variants in the gene. And on the Y is all of the common missense and loss of function variants. So you can predict the expected amount of common functional variation in gene given this X value. And what they're highlighting here in red are genes that have less common functional variation than predicted. So this is the regression across everything. And some of these genes down here just don't have that many common functional variants. And I'm sure you can recognize a few of these. These are definitely disease-associated genes, and it's a useful way of highlighting genes that appear to be intolerant to mutation. In Nomad, we took actually this latter approach to depletion of rare variants compared to expectation. And the logic behind it is that, let's say you have a tolerant gene, a gene that can be mutated, and an intolerant or a constraint gene doesn't like to be mutated. When mutations arise, potentially in a tolerant gene, one of them will be removed from the population by selection, but a constrained gene, a lot of the mutations will lead to a deleterious phenotype and therefore be removed from the population. So what we see over time and grouping individuals together is that a tolerant gene will have a number of these rare variants, these rare mutations and differences, but a constrained gene will be lacking that expected amount of variation. You will not see these rare deleterious variants. So we have to have an expectation here, and we have developed mutational models to be able to do this. So these are based on sequence context pretty much alone, some corrections for regional factors or sequencing depth of coverage. But what we want to do here is show you that these work. So here we're comparing, again, the observed minor allele frequency less than 0.1%, so observed rare variants in a gene compared to the expectations from a mutational model. When we look at synonymous variants, for example, in Nomad V4, we see a very tight correlation. So each of these dots is a gene, and we can see the expected number of synonymous variants and the observed number of synonymous variants. And that is a very tight, high correlation. So our mutational model, we trust, can predict these mutations. So we then apply it to missense and loss of function. And for missense, we are still seeing a pretty high correlation. Overall, the vast majority of genes have most of their missense variation. There is a little bit of a depletion falling off of this line, but really where we see that depletion is when we look at loss of function variation. So nearly all genes have less loss of function variation than we would predict from this selection neutral model, right? It's just based on the mutability. So we suggest that this loss of function depletion indicates the action of selection. So this depletion we wanted to quantify in a couple of different ways. So for example, we can measure with a more dichotomous metric. This is an older one called PLI, or the probability of loss of function intolerance. It's very much a yes, no. So PLI goes from 0 to 1. We arbitrarily say that greater than 0.9 indicates a constrained group. This is about 3,000, 4,000 genes across the genome. This is the distribution here, so it is very dichotomous. And a lot of the ones in the middle are on the smaller side. By contrast, more recently, we developed LUF, or the loss of function observed over expected upper bound fraction. This was designed to be a more continuous metric. And this distribution I have here is from the V2 dataset, but we see something very similar when we apply this to Nomad V4. So as you get to lower LUF values, this means that you are more constrained. And the reason we want to put an upper bound on this is, again, some of these smaller genes, if you see 0 out of 2, that's a little bit different than seeing 0 out of 20. And so we wanted to just put a little bit of confidence on that, but that's sort of the logic here. So when we take, for example, this LUF value, the loss of function observed over expected upper bound fraction, we do see that the most constrained LUF deciles here, the first and second in particular, are enriched for haploinsufficient genes, which is a nice check. Olfactory genes, those olfactory receptors that seem to be able to be mutated in humans with no obvious negative phenotypic consequences for the most part, those are enriched in the least constrained LUF decile. So again, a nice quality check. And autosomal recessive genes are spread across the whole spectrum. So these constraint scores here that we're talking about, they're really getting after this sort of heterozygous dominant signal. So these recessive disease genes are obviously important for human disease, but they don't seem to have that impact on the heterozygous side, or not completely, and we do see them spread here. So it's nice to see that these well-known gene sets sort of interact as we anticipate. But when we turn to looking at, for example, Denovo, newly arising variants in cases with intellectual disability or severe developmental disorders compared to controls, we also see that these constrained genes or these deciles help us pull out that signal. So really, when comparing the rate of Denovo variance in these cases compared to controls, usually neurotypical siblings from autism trios, we see that most of this enrichment case to control is in this lowest decile. So the reason we like to highlight this is that it's not just useful theoretically, it is useful also in identifying disease genes or helping prioritize variants. So if you see, for example, a new Denovo variant, a Denovo loss of function variant, in a gene that's massively depleted of loss of function variants in the general population, that is one way to upweight. So we can, of course, combine allele frequency and selective constraint together. This is one of the most powerful ways of doing this. So in this 2017 paper, what was done is they were looking at, again, these de novo loss of function variants in intellectual disability, developmental delay, and autism patients, and comparing them to, again, those unaffected siblings from the autism families. And there's this well-known excess of about 2 in neurodevelopmental disorder individuals. So we see in blue for the developmental disorders, this is 2.6 higher for a rate compared to the unaffected siblings. It's about 1.8 for individuals with autism. But when taking 2 pieces of information into account, almost completely cleans up the signal. So if that de novo loss of function was seen in a population database, so this is exact, but the same thing holds for Nomad or UK Biobank, or if that de novo loss of function variant was in a gene that was not considered loss of function intolerant, there's actually no significant differences in the rates between cases and controls. All of that signal, almost all of that signal, is for those de novo loss of functions that have never been seen before and to interrupt these loss of function constraint genes. So again, we could have used the LUF deciles here. We use PLI because this is a slightly older paper, but this is a one way that allele frequency in these population databases, as well as constraint, can really help prioritize and resolve some of the signal. So I've been talking about loss of function because it's a little bit more straightforward. Obviously, there are more challenging classes of variation. Missense variants, for example, are much more complicated, both because the specific amino acid substitution matters, as well as its location within the gene or the protein. So we would expect here that instead of an entire gene being intolerant of missense variants, only some sections of them might be. And there have been a number of different approaches to this over the last couple of years. And there are three primary ways of going about it. So the first is linear. So this is looking in DNA space, or like kind of horizontal. So here it's trying to identify in DNA space, like these two exons are depleted of variation and these are not. So there have been a handful of papers here. This figure is from the constrained coding regions from Hebrilla et al. In 20 in Nature Genetics. The second approach is using homologous regions across proteins, and this is more vertical. So this, for example, and this figure is from Zhang et al. In Genome Medicine, is looking at ion channel families from across SCN1A, SCN2A, for example, lining up those positions across all of these genes to identify for a given domain position what seems to be tolerated or not. So this is kind of stacking the vertical. And the last is structural. So there are a few papers from before AlphaFold, but obviously AlphaFold 2 and AlphaFold 3 make this much more powerful. So using the 3D structural space. So this is coming from Lee et al. In Nature Communications, and this is an 8 angstrom ball around a given residue, and you can use that to identify constrained approaches. So in Nomad, we've been doing this linear approach. We've released it based on Nomad V2. We have found that about 30% of transcripts have a significant difference in regional difference in the rare missense observed to expected fraction. So for example, in this little play case here, maybe these two exons are very depleted. They have only 25%, and the rest have most of it. For a real example here, this is KCNQ1. So we see this first part of the protein has only 66% of its expected missense variation. This end bit is about 60%. When we overlay the ClinVar pathogenic variants, we see that they are nicely aligning with what's depleted. And the reason that this is a nice approach is that we're just using the population data. There's no information here about what's pathogenic influencing these. Obviously, this is a nice cherry-picked example where things align well, but we do see that the regions within the human exome that are depleted of missense variation are enriched for ClinVar pathogenic variants, and they're also enriched for the Denovo missense variants we see in patients with neurodevelopmental disorders. So exactly those cases I was talking about a few slides ago, we also see that here. So this is currently available on Nomad V2, which is 125,000 or so individuals. It's currently being applied to Nomad V4 with a hope to being released later this year. We do also have a couple of other constraint metrics that I won't go into in too much detail in terms of time. So based on the 76,000 genomes, we have NOCCI, or the genomic non-coding constraint of haploinsufficient variation. So this is a z-score based on 1 kb windows around the non-coding regions. And this is, for example, as a visual of the score, so you can see that on the browser. We also recently added mitochondrial constraint metrics developed by Nicole Lake and al. So if you're looking at mitochondrial genes, there's observed and expected SNVs. We've also pulled in regional missense constraint metrics from Nicole's paper. And if you're interested in these, I suggest you check out the publications because they have a lot more information about what's going on there. Right, so we've been able to use this population resource to filter variants. We've also identified genes that are intolerant to mutation generally. The last bit I'll tell you about is statistical phasing. So this is also looking at patterns of variation, but it's using them to apply to recessive diseases. So the knowledge of the phase of two variants is really important for recessive disease diagnosis. So if you see, for example, that an individual has two damaging variants in the same gene, let's say two loss of functions in the same gene, you really care, if it's a recessive condition, whether it's in the same haplotype in cis or whether it's an opposite haplotypes in trans. And that's because if it's in cis, it's not going to lead to disease, where if it's in trans, it could. And these are the situations we're looking for. So obviously, if you sequence father, mother, and child, you have a better chance of being able to phase things. But when you're looking at short read exome sequencing data, this is extremely hard to do. Exons are quite far away from each other, so you can't really link them. If you don't have parental information, that's another lost piece of the puzzle. So the idea behind the approach that we have here is that if a variant seems to be cis in the population, it's probably in cis in any given individual. And if these variant pairs are in trans in the population, they're probably in trans in a given individual. So this is a paper that came out early 2024, and that was led by Mike and Laurent. So we'll walk through the logic here. So we used an expectation maximization algorithm, actually from a fairly old paper, to infer phase. So as an example here, if we see two variants in trans, they're going to be in trans in the whole population. Essentially, we're never going to see them in the same individuals. And so we can tabulate genotype counts. We use these to estimate haplotype frequencies, and then we can calculate the probability of these two variants being in trans. And similarly in cis, we'll probably always see these variants traveling together in the population. This is obviously a vast oversimplification, but this p-trans metric that we make at the end of the day is the probability of inheriting each of the haplotypes that contain only one of these two variants. So this sort of scenario here. So we have done this both on the global Nomad population as well as on individual genomic ancestry groups within Nomad. And we see that the inferred phase actually performs quite well. So we had about 5,000 trios that were jointly called with Nomad but not released, that we used as a truth set here, where we could use the parental information to do this gold standard phasing by transmission, right? We know the parental information, and then we compare all of the variants. We try to predict phase from our particular performance, and we do see that overall we have fairly high accuracy. And this method, I should say, does require that both variants are present in Nomad to predict phase. In this truth set, that was about 91% of the variants. This is not necessarily the perfect example, given that they were jointly called with the data set, but I'll have another example here. So there are about 8 or 9% that weren't phased because they weren't present in Nomad. We do see that there's another chunk that get an intermediate p-trans value, so they don't clearly seem to be in trans or in cis. But for those that we can phase well, we are quite accurate. And overall, these are sort of these accuracy measures. You can see that this works even to fairly rare variant values. Where we start to fall apart a little bit, where the model struggles, is two variants that are in cis that have very different allele frequencies. So if one of them is very rare and one of them is a little bit more common, that becomes a little bit harder. So just to be upfront about where this works well and where it doesn't, but these accuracies are still pretty high. So for an application to a disease cohort, we are using patients from the Broad Center for Mendelian Genetics that have a confident or strong candidate recessive disease diagnosis, and they're all presumed to be compound heterozygous, so we presume that the variants are in trans. So we started with about 627 variant pairs, of which we could phase a little over half of those, 293. And of those 293, we predict that 281 of them, or 96%, are in trans. So we're matching the prediction from the cohort. 7 we predict to be in cis, and 5 had indeterminate values. So a handful we can't predict. So for these 7 that we are apparently wrong for, 6 of those were only proband sequencing. So we actually don't have the parents there. So these could be an incorrect phase prediction. We'll be upfront about that, but it also could be an incorrect diagnosis. So we did let our collaborators know here to go back and check. There was one that came from a full trio sequence, and so we were just wrong about that particular individual. But just again to reinforce that for the 50% or so of cases that we applied this to, it was helpful in the vast majority of them. So we also wanted to be able to provide a little bit more of a reference for individuals. So we are counting rare genes that, we are counting up in the same gene how many individuals have 2 rare damaging variants. So we've done this previously looking at homozygous loss of function variants, sort of complete knockouts, but compound heterozygotes is something that is less available. I should note there was some nice work from UK Biobank out of Oxford looking at this. So we counted individuals with 2 rare variants in the same gene, and we stratified by predicted phase, allele frequency, and consequence. So we used synonymous missense, Revel missense categories based on ClinGen recommendations, and then loss of function over on the right. And just to shorten this, we did not find very many genes that have compound heterozygous carriers of rare deleterious variants. So looking at allele frequency less than 1%, for loss of function, really there's not a lot going on over there in compound het in this kind of peachy color. The blue is the homozygous variant. So you see that across all allele frequencies in all classes, but really not a lot for loss of function. I should be upfront though, a limitation here is that since we use Nomad to train the model, we can only predict in trans variants down to an allele frequency about 0.1%. So we do struggle a little bit with some of the most rare variants, but what we saw actually in that other paper from UK Biobank, there's not that many kind of compound heterozygous variants in sort of constrained genes floating around either. But it's useful to look into and to quantify. So we did have 28 genes, right, 28, with predicted compound heterozygous loss of function variants in our data set. And we did a little bit of curation here of all of these. So there were 25 of those were non-disease genes, 2 were dominant disease genes, and 1 was recessive. And we had our Muriel and Emily do some expert curation, the loss of function framework I mentioned a little bit earlier. And essentially, all of the loss of function variant pairs in these disease genes were ruled out. One of the 2 at least looked like it wasn't a real loss of function. Of the 25 non-disease genes, there were 8 loss of function pairs in 7 of those genes that looked like they might actually both be true loss of functions. Those genes are listed here, but again, they're currently not disease-associated genes, which is potentially why you could see them in a reference population. So we've released this. It's only on Nomad 2.1 right now, where we're slowly moving things over to V4, but you can look up estimates for over 5 billion pairs of variants across all of the genes. And as always, I should say, we have a lot of information available on the downloads page, so you can access most of this yourself. So just to wrap up with what's coming in the future, V5, we're aiming for later this year or early next year. That should include diverse genomes coming in from the All of Us dataset. It's over 400,000, potentially over 500,000. So that will join the genomes that we currently have. So this will be a much, much larger and more ancestrally diverse dataset. And then for V6, we're shifting to more of a federated model. So the idea here is that we know that there are datasets being generated around the world that potentially cannot move countries due to regional specific restrictions. So we've set up a network and we're working with teams already to process data according to the Nomad approaches. We're going to help a little bit stand up browsers around the world. And the goal then is to aggregate those allele frequencies back into the primary resource to get improved allele frequency information for community use, filtering allele frequency, and then improved constraint analyses. So these have, this project has just started. If you are interested in joining the Federated Nomad Network, please reach out. We're always looking for more people. So just to wrap up, we do think it's quite important to have these large databases of genomic data coming from reference population individuals, specifically those with diverse genetic ancestry variants. Diverse genetic ancestry. And we can use this population data to filter variants, not just based on frequency or some disease-associated frequency, but also to use the patterns of variation to identify genes that are under selective constraint or to potentially predict phase in exome sequence samples. And of course, variant curation is always something to keep in the back of your mind. If something looks interesting, you might want to check out how much you believe it. And with that, I'd like to acknowledge the core Nomad team. The individuals with pictures over on the left make up that team, but obviously many, many people contribute to this. So data contributors and the number of individuals that have aided in this platform off to the right, and we have more information on our page. And with that, I'm happy to take questions.
Kaitlin, thank you so much. That was absolutely wonderful. So if people would like to ask questions, they'll see on their screen there's a Q&A button just at the bottom. So if you click on that and then type away your question, and then I will moderate that to Kaitlin. So please do start tapping away. I have got sort of chairs sort of opportunity just to ask a couple of things, if that's okay. Kaitlin, it was really interesting to hear at the beginning how you've really sort of put a lot of emphasis into looking at the data emerging from different ancestry groups and the sort of flagging the value of that, and how in some ways increasing the numbers significantly from European ancestry doesn't provide lots of new information, whereas you saw the really relatively small increases in population, release huge amounts of information, particularly from the African and Middle Eastern populations. Do you see, is there a way of calculating the numbers of individuals you will need from the Middle East? And when we talk of Africa, I mean, Africa is an enormous continent and there's so much variation. And have you got a sense that actually in some parts of Africa, there are sequencing programs going on which are helping to understand that variation, whereas other bits of Africa that's just not the case at all?
Yeah, so to tackle the first question, that filter, that plot I was showing, I was looking at variance with an allele frequency greater than 0.1%. So essentially, as soon as you hit about 10,000 individuals from certain populations, you're starting to saturate in that space. We could shift that lower, though, because we know that disease-associated variants are much more rare than that. So we do think that there's power still in sampling from the European population, but in some sense, you get more bang for your buck, to be colloquial, out of
Yeah,
sequencing individuals from populations that aren't currently well represented. So
yeah,
you're right. Middle Eastern in particular is missing. Oceania, we don't have really at all on
yeah,
those plots. Southeast Asia. In terms of Africa, I'll take a moment to say, actually, most of the African genetic ancestry that's being represented is coming from individuals from West Africa. And that's
yeah,
because almost all of these individuals being sampled from the US and the UK, and they are historically were being brought from West Africa. So
yeah.
South African genetic ancestry and East African genetic ancestry in particular are fairly depleted. We are working with individuals, I believe H3 Africa, in the federated nomad approach to try to, for example, get better representation from those populations, because it does influence the clinical utility of these databases. If you are coming from East African population, there may be population-specific variation that's not in our database, so it looks rare, so you can't filter it out in a clinical group. So there are, I think DDD Africa is collecting patients there for neurodevelopmental disorders,
Mhm.
NeuroDev, NeuroGap. I know of a number of studies that have been started. Obviously, major ones like H3 Africa should be, they're doing an excellent job. And
Yeah.
the hope is to empower people to stand up their own work and share globally as well, because that's going to help people around the world.
Yeah, no, that's fantastic, and it's great to hear about the federated plans for V6, which we can talk about maybe in a second. So I'm going to move to some of the questions that people have started popping into the Q&A section. So the first one's from Katrin Janssens. Katrin's asking, when will the loss of function curation be available in Nomad version 4? Have you got a
The
sense?
team is working. The team is working on that. It's actually quite a lot. The real question is whether you can lift over the curation from V2 to V4. The major challenge here is that there was a genome build change. So it's moving from 37 to 38. So some of these mapping issues or transcript issues that we would have flagged in V2, we're not sure actually hold in V4. So there is some work being done on that, but it is a slow moving process. And I believe that there's a curated set that they're going to be of genes that they're going to be working through first. So it's underway, but I don't like giving timelines.
Yeah, sure, sure. No, that's fully understandable. So Chaka Louis has asked, and everyone's saying what a great presentation, but has asked, can you use public databases like Nomad to understand where digenic inheritance might be relevant? It strikes me as complicated, but you've probably given that more thought than me.
Right. So I would say that I really like Nomad. I'm biased, but a major con of Nomad is we're not allowed to share individual level data. So
Yeah.
we don't have phenotype data available, and we actually can't even really release genetic information on individuals. The 1,000 Genomes Project samples and the HGDP samples that are in our data set, you can find that, but that's
Mhm.
because they're from other resources. So this select statistical phasing work that we do is sort of the most that we've been able to release, and that's just saying that there's a certain number of individuals that have two rare variants in trans or in cis with each other. But digenic would require us releasing that across genes, since that's multiple genes involved. So actually, currently Nomad does not have the capability to do that. I would suggest looking at UK Biobank or all of this, where you have access to individual level data as well as phenotype data, so you can really dig into that
Yeah,
question. But
yeah.
it's a hard one.
It is. It is. That's a very good point. So Gideon Wiafe has asked, when you're thinking about Denovo variants and being confident that you've identified one, is that going to be possible where you've only got sequence data from an individual, where getting trio data is a challenge because of resource limitations, by inferring from population data sets?
Right. So that's a good question. I would say it's extremely hard to infer that something's de novo. There are certain genes that are extremely depleted of certain variation in the general population and really enriched. I'll take, for example, DDX3X, which is associated with severe developmental disorder. DDX3X has a lot of de novo loss of function variance in, for example, the Deciphering Developmental Disorders study, and we really don't see that many loss of function variants in the general population. So if you see a loss of function variant there, you could probably think that it's likely to be de novo, but establishing that it actually is using population data is challenging because you would need to take into account, for example, how mutable the specific base is. There may be a way to do it, but we don't necessarily currently have a strong setup for that. Even getting one of the two parents helps in that scenario because it moves the prior probability that it's de novo around. But if you're using this in a clinical setting, I am hesitant to suggest that you could fully use population data just because the bar for making a clinical judgment based on that information is high.
Yeah, no, fully understandable. If you've got a good candidate, you probably want to test the parents just for that variant, just to and be confident that it really is de novo. But yeah, it is challenging where lots of places it's very difficult to undertake trio analysis because it's expensive. Can I just ask? I thought it was really interesting where you were able to look at that group where you saw the loss of function variants in the haploinsufficient genes, and you highlighted that the majority of those, 70%, are not true loss of function for a whole range of reasons. But that still leaves 30% that possibly are, which is quite interesting. I mean, I know there's limitations of what phenotype data you've got access to know whether those individuals truly have a phenotype, but there could be sort of other mechanisms that I don't know. Is it going to be possible to explore some rescue from the other allele of overexpression or something like that that could allow us to sort of think that there's some compensation going on that means that we're not seeing those phenotypes in relevant individuals?
Right. So I know that there is a lot of interest in the kind of general penetrance question, and Caroline Wright's team has had a few papers on this. For example, looking at pathogenic variants in associated developmental disorder, associated genes in individuals in UK Biobank, and showing that some of these carriers do have a shift in sort of related phenotypes. They might not necessarily, like some of them do have ICD codes associated with developmental disorders or learning disabilities, but you do see potentially a shift also in maybe fluid intelligence scores. I don't want to misquote the paper, so I'll just say that there's a nice paper looking into that specifically. But that again is where you would need sort of the access to the phenotype data. You're absolutely right that it could be overexpression, it could be some compensation mechanism, it could be, there's a lot in the genetic background that we're not looking into. And so that's why, to some extent, we know that a few things are going to slip through, but this 30% would be an interesting one to dive into. And so finding carriers in all of us or in UK Biobank and one of these other cohorts would be the way to go about that.
Yeah, no, that's really helpful. And it's not the expectation that Nomad can do everything, but it creates a wonderful resource to start thinking about some of the other questions that we have. So Peer Arts asks, lots of groups are now generating long read sequence data. Are there any plans to incorporate that into a future Nomad reference set?
Yes. Yes, we have some long read data that hopefully will be available later this year. I actually believe a lot of it is also coming from the All of Us project, but we're working quite closely with their teams. That is the plan to have that. Long reads are a much smaller resource, so we're aware that, for example, if you're doing allele frequencies and coding regions, the V4 dataset is still going to be more powerful, but long reads give much better insights into complex structural variants and certain regions of the genome. And that's why we understand that it's an important thing to get out there. So yeah, it's on the roadmap. The hope is to have that available soon, but timelines are scary, because then that means I'm signing up our poor production team to get all of this out. A lot on their plate.
Well, it's great that there's still lots of exciting things to do. So one question come through from one of the attendees that sort of, as there are a number of individuals with clinical phenotypes in Nomad 4, is that raise concerns that actually classification of whether a rare variant is pathogenic is more challenging?
Certainly it's more challenging. I think the problem here is that we did not do a good job with the earlier releases with making it clear that there's always been individuals with some condition in the data set. Like, no one is perfectly healthy. Everyone has so for example, you might be a control for schizophrenia, but you might not be a control for a cholesterol study. And so who defines what's healthy enough to be included here? Yes, we have seen an increase, for example, in some pathogenic variants, that Delta F508 variant in CFTR, we see at a much higher rate. And we tracked it down. It's coming in from a biobank that had a lung clinic. And so that's a particular case where we were able to figure it out. However, there is reduced penetrance, and people sometimes do get into things like UK Biobank with a KMT2D loss of function. And so they are the general population, if that's what you're sampling from. I'm aware that it provides some additional challenges, but we've always been clear that we don't think the presence of it once in the database should be used as ruling something out, just because there are sequencing errors. And sometimes we could see something in our database that's not real.
Yeah, absolutely. And finally, with the federated database, I mean, I think that's incredibly exciting, because I know that people have always expressed this concern and sort of frustration and disappointment that we've not been able to share some of the great resources that are being generated in different parts of the world. So are you getting lots of positivity that that's going to be a reality? I mean, of course, it will take time, and there'll be various agreements that need to be put in place. But is your sense that actually people are converging around that?
Yeah, we've had a number of calls already, and we have a handful of groups that are on the closer side for standing up their own browsers and contributing. I don't necessarily want to call them out specifically on this webinar that's going on YouTube. But yeah, we have a handful of groups that are closer getting ready. I think the major every group has a different kind of challenge. So some teams, it's annotating the data set, or like doing the QC the same way. Other groups, it's building the browser, and we're trying to establish that and get it close. But there has been a lot of excitement about this, and we do have a number of teams that should hopefully be ready to release again, maybe sometime in 2026.
That's fantastic. Kaitlin, thank you so much for your time this afternoon, for such a wonderful presentation. It's the culmination and ongoing efforts of you with a large team and an enormous amount of work that really does support the whole genomics community. So thank you from all of us for all your efforts. And for such a great presentation today. Next month, we have Siddharth Banker from Manchester presenting. So it'll be the same time, same place, the last Wednesday of the month, 4 p.m. Central European time. And we look forward to seeing you then. If you didn't catch all of Kaitlin's presentation, or you want to tell your colleagues about how brilliant it was, it should be on the ESHG YouTube site very soon. So thanks for joining us, and thanks very much, Kaitlin.
Thank you.